So very good afternoon everyone. I am Dr. Satyabrata, MDDM clinical pharmacologist, and today I am going to discuss on mechanisms and applications of apoptosis based and molecular targeted therapies. Coming to introduction, what is apoptosis? Apoptosis is a programmed cell death and orderly and efficient removal of damaged cells following DNA damage or during development. It is very complex and involves many signaling pathways. It is triggered through caspase mediated intrinsic and extrinsic pathways. There is balance between pro-apoptotic and anti-apoptotic regulators that is important key point in apoptosis. The dysregulation of apoptosis is a symptom in a wide variety of diseases. There is accelerated apoptosis. It is found in infertility, immunodeficiency, and acute and chronic degenerative diseases. And delayed or inhibited apoptosis is present in cancer and autoimmunity. This is a picture showing intrinsic and extrinsic apoptotic pathways. The intrinsic mitochondrial and the extrinsic ligands of the death receptors, cell death pathways, and their convergence through TBID. And in intrinsic pathway, stress signal will affect the BCL2, BCLXL, and TBID backs back, and then the apoptosome, APAP1 and procaspase 9 will be produced. Procaspase 9 will activate the caspase 9, then procaspase 3, 6, 7 will be activated, and BID and also procaspase 3, 6, 7 then activate the caspase 8, 10, and caspase 8, 10 will activate the BID, and then the intrinsic pathway continues. And in the extrinsic pathway, ligand when bind to the death receptor, that is FAD, TRAD, and procaspase 8, 10. This is known as DISC disc and disc complex, and it will activate caspase 8, 10, then procaspase 3, 6, 7 activated, and at last procaspase 3, 6, 7 will activate the caspase 3, 6, 7, and cleavage of critical cellular substrates, then apoptosis occurs. This is the mechanism of apoptosis, intrinsic extrinsic pathway. So this is another picture diagram showing the pathway of intrinsic apoptosis. BH3 only proteins are upregulated in response to apoptotic stress. They activate BAX BCL2 associated X protein and BAC BCL2 homologous antagonist killer, which oligomerize and results in mitochondrial membrane permeabilization. Cytochrome C, SMAC, the second mitochondria derived activator of caspase, and omni are released, and the apoptosome is formed from procaspase 9 and DATP, cytochrome C, and APAP1. Caspases are then activated and begin to cleave cellular proteins, resulting in apoptosis. Arrows represent activation and T-bars represent inhibition. So, apoptotic stress, DNA damage, uncontrolled proliferation, etc. BH3 only proteins, and BH3 only protein activate the BAC oligomerization in the mitochondrial membrane permeabilization, and then the cytochrome C will be released, SMAC, OMI will be released, and then caspase activation, apoptosome activation, cytochrome C cause apoptosome activation, then caspase activation, then cell death, and XIAP molecule involved in this, and anti-apoptotic BCL2 members are also involved. These are some diagram that is related to apoptosis, intrinsic apoptosis. So this is another diagram showing the extrinsic pathway begins with a death ligand docking on a death receptor. An adapter protein binds to the receptor disc, that is death inducing signaling complex is formed from the adapter protein and procaspase 8 and 1 alpha. Then caspase 8 becomes activated, which activates caspase 3, 6 and 7 and BID BS3 interacting domain death agonist. BID goes on to activate BAX and BAC, which activates the intrinsic pathway. Caspase 3, 6, 7 are the executioner caspases that result in cell death. Arrows represent activation and T bars represent inhibition. So you can see the death receptors TNFR1, FAS and DRS, and on the other hand there is death ligand TNF alpha, FAS-L and TREL. Then adapters FADD, TRAD. Then there is formation disc, death inducing signaling complex, and it will activate the caspase 8, then caspase 3, 6, 7, then apoptosis and intrinsic pathway, and also activate the BID. Caspase 8 to activate the BID and intrinsic pathway continues, and SMAC inhibit the XIAP. XIAP will inhibit the caspase 3, 6, 7 and FLIP will inhibit the caspase 8. So this is a diagram showing the extrinsic pathway and also intrinsic pathway of the apoptosis. Then this is another diagram showing the function of inhibitors of apoptosis proteins, IAPs. IAPs are often overexpressed in cancer and they have the ability to bind and inactivate caspase 9 and 3. The activities of IAPs, inhibitors of apoptosis proteins, on the other hand, may be suppressed by mitochondrial proteins such as Omni-HTRAZ and SMAC-Diablo released into the cytosol during apoptosis. So the caspase 3 will cause apoptosis. IAP inhibitors of apoptosis proteins, caspase 3 lead to caspase 9 and caspase 9 activate the caspase 3 and lead to apoptosis, but SMAC-Diablo will inhibit IAP and Omni-HTRA2 will inhibit IAP. And this is all about this diagram. Next, this is another diagram showing the mechanisms leading to deregulation of apoptosis. Schematic represents of the different ways through which both the extrinsic and intrinsic apoptotic pathways may be altered, resulting in reduction of apoptosis or acquisition of apoptosis resistance. So, deregulated apoptosis cause cancer development and chemoresistance, and overexpression of anti-apoptotic proteins, downregulation of apoptotic proteins. There is unbalance of BCL2 family members, and overexpression of IAP proteins, downregulation of caspases, impaired death receptor signaling, impaired WTP53 function, and this is a complex mechanism of deregulation of apoptosis. So this is a table showing the summary of plant derived compounds and their mechanism for inducing apoptosis. Alloy imodine found in Rheum pulmantum induces cytochrome C release, and black cohosh Actaia racemosa will activate caspases. Curcumin, that is from turmeric, found in turmeric, inhibits BCL2 and XIAP, and Genstein soybeans cell cycle arrest activation, and graviola among some plant name is there. Inhibits BCL2, activates BAX. So there are some quercetin found in bark of many plants, modulating cell cycle regulators to arrest the cell cycle. You can see lots of articles about this plant products. Then this is a table showing a complex table showing apoptosis based therapeutics in clinical trials or in preclinical stage, and death receptor, trail receptors principle, and lots of things are there that is in clinical trial phase 1, and CD95 fast molecular target, it is also preclinical stage. TNF, recombinant TNF alpha, and approved in isolated perfusion, limb perfusion therapy in melanoma. Humira, adalimumab, that is TNF alpha, action TNF alpha, FDA approved in rheumatoid arthritis. Infliximab, FDA approved in rheumatoid arthritis, Crohn's disease. Then there are lots of CDP 571 phase 3 for rheumatoid arthritis stopped, and these are some example of apoptosis based therapy. U. Fisher and K. Schultz Ostop. So these are some complex mechanism of apoptosis, clinical trials on apoptosis. Then the caspages, pan caspase, IDN 6556. This is in phase 2, started for chronic HCV infection, phase 2 open for HBV infection and ischemia reperfusion, injury of liver transplants. Then IDN 6734, phase 1 acute myocardial infarction. Then MX2060 derivatives found in a cell-based caspase screen growth inhibition in a rat tumor model, preclinical stage. So this is also some examples of clinical trials, preclinical trials and studies in apoptosis based therapy, U. Fisher and K. Schultz Ostop. This is the reference, you can go through that. So this is also, there is caspase 1, that is also in phase 2 rheumatoid arthritis patients showed anti-inflammatory effects. Caspase 3 preclinical stage, caspase 6 in preclinical, and experimental effects of caspase 3 is growth inhibition of HOR2 positive tumors in a mouse xenograft model. And this is the reference is apoptosis based therapy, U. Fisher and K. Schultz Ostop. You can go through the articles about to learn more about this. So these are also examples, IAPs, MAC, and BIR3 antagonists. These are in preclinical trial, preclinical study. Then MBEN, it is in preclinical study. You can go through apoptosis based therapy, U. Fisher and K. Schultz Ostop to learn more about all these things. So then BCL2 proteins, anti-apoptotic BCL2 members, BCL2 blockers are in preclinical studies. Then lots of others are also in preclinical studies. So you can go through apoptosis based therapy, U. Fisher and K. Schultz Ostop to learn more about these things. So this is also Genasense in phase 3 FDA fast track status for melanoma, multiple myeloma, chronic lymphocyte leukemia, and phase 3 for non-small cell lung cancer, phase 2 for hormone refractory prostate cancer. Then others like SAHBs in preclinical, BH3Is in preclinical study. And you can go through apoptosis based therapy, U. Fisher and K. Schultz Ostop to know more about all these things. And this is also a long example, amifostine, established as a chemo and radio protector with complex mechanism of actions, and prima 1 preclinical might serve as lead compound These are lots of examples of apoptosis based therapies. You can go through apoptosis based therapies, U. Fisher and K. Schul, Osthoff, to learn more about these that that are in the preclinical or clinical stages, clinical phase, trials. You can go through to learn more and more. And this is also another examples of those in preclinical studies. Chalcones in preclinical, nucleins in preclinical lead compounds, HPV-E6 binding peptide, aplamers in preclinical study. So you should go through apoptosis based therapy, U. Fisher and K. Schul, Osthoff, to know more about all these things. So then coming to novel therapeutic target agents targeting apoptosis. Novel therapeutic agents targeting apoptosis, that is oblimersen sodium, that is used in AML, acute myeloid leukemia. Troglitazone, that is used in advanced prostate cancer. Fenretinide, used in rhabdomyosarcoma and ovarian carcinoma. Flavopiridol, that is AML, acute myeloid leukemia, small cell lung cancer, SCLC. Gosipol, SCLC, small cell lung cancer, NSCLC, non-small cell lung cancer. Then R7112, advanced solid tumors. APR246 in malignancies, hormone refractory prostate cancer. Dulanormin, non-Hodgkin's lymphoma. Tigatuzumab in breast cancer. Decitabine in caspase 8 promoters booster. Coming to summary, apoptosis is a cellular procedure regulated by diverse groups of regulatory molecules in which activation of caspase plays a crucial role. And depending on the molecular target, different strategies are being employed. And fundamental features of human cancers is evasion of apoptosis. So, this leading to tumor progression. Evasion of apoptosis will lead to tumor progression and resistance to cancer treatment. So, biologicals including death ligands or agonistic and antagonistic antibodies that inhibit or trigger death receptor signaling have proven efficacy in various animal models. And among all the apoptosis based drug target strategies that target caspases are at the forefront for blocking apoptosis in numerous diseases. So proof of concept data have been obtained in several experimental models revealing that blockade of apoptosis by means of caspase inhibitors is a validated therapeutic strategy. Many compounds developed to modulate apoptosis are only micromolar inhibitors. Small molecule drugs or peptides like caspase inhibitors have to be optimized to readily penetrate the cells. Drugs that selectively target different components of apoptosis are in clinical trials. Combination of these novel agents with traditional chemotherapy seems to be well tolerated and benefit for patients. So these are the references. You can go through these references and search in the Google and you will find the articles and you can learn more and more about this topic from the articles. And also if there are some guidelines in foreign countries or in India, you can search for that and also learn more and more. And please like and subscribe my channel and completely watch all these videos so that you can learn a lot from these videos. And you can also search in the Google for the articles and guidelines and you learn more and more. And the more you learn, the more you will gain knowledge and you can help others also. So please like and subscribe my channels, share, like, and also you can advise for next topic also Also you can comment in the comment box there after uploading the video, and I will try my best to answer all the questions. And that's all for today. Thank you very much for watching and listening to me. Jai Jagannath. Dhanyabad.